Shifan, and in this video we will be going through the last 10 questions from the G.C.E level biology MCQ paper 2023. On our previous videos we went through the first 40 questions from this paper, right? And now let's go through the multiple response questions, which are the last 10 questions. Unlike the first 40 questions, these last 10 questions can be a bit more tricky, right? But if we read it carefully and understand each statement, we can find the right answers. Okay, now let's go through question 41. Which of the following statements regarding nucleotides are correct? Now, what are nucleotides? Now, nucleotides are molecules that consist of a phosphate group, sugar, and a nitrogenous base. So this nitrogenous base can vary. They can be adenine, thymine, guanine, or cytosine, right? And the well-known nucleotides are DNA and RNA. But other than that, the nucleotides that are really important for the body are NAD+, FAD, NADP+, and also ATP. These are all nucleotides. So that's something I want you guys to understand before we go through the answers. Now, first statement says NADP+ functions as an electron carrier and an oxidizing agent. Now, if you take molecules such as the oxidized forms of molecules such as NADP+, okay, NAD+, and also FAD, okay. Now, these are all oxidized form of the molecule, which means they can gain electrons, or in other words, they can accept electrons, right? Because they are all oxidizing agents. These molecules, they can accept electrons, they can carry electrons to another place. So when they accept electrons, these become the reduced form, right? NADP+ becomes NADPH, NAD+ becomes NADH, and FAD becomes FADH2, right? In other words, when they accept electrons, they gain energy, and these molecules are just like batteries without charge, right? But if they get electrons, they get charge. So that's a way to understand that. Okay, now let's go through the first statement. NAD+ functions as an electron carrier and an oxidizing agent. As I said, oxidizing agent are anything that can accept electrons. But if you take NADP+, they can accept electrons. For that reason, they are oxidizing agents. Now, when they accept electrons, they usually carry the electrons to another place. In the case of NADP+, where they are involved, NADP+ is usually involved in photosynthesis, right? Photosynthesis. In the light dependent reaction, NADP+ is reduced to NADH. Let me explain it. Okay, wait. Now, in photosynthesis, what happens when photon strikes the chloroplast? Electrons are excited. Now, finally, these electrons are accepted by NADP+. And when they accept electrons, they become NADPH. Now, this NADPH moves into the Calvin cycle and is used for the production of glucose, right? So they are also an electron carrier. They carry electron from light dependent reaction to the Calvin cycle. So they are both an electron carrier and oxidizing agent. That statement is correct. Now let's go through the second statement. FAD functions as an electron carrier and a reducing agent. As NADP+, FAD is also an electron carrier. Okay, but where is FAD is involved? FAD is involved in the citric acid cycle. If you have learned aerobic respiration, you know they have three processes: glycolysis, citric acid cycle, and the electron transport chain. In citric acid cycle, what happens? FAD+ gains electrons and becomes FADH2. This FADH2 moves into the electron transport chain where they can be used to make ATP. So they are an electron carrier, but are they a reducing agent? FAD, as I told, they accept electrons, right? When they accept electrons, anything that accept electrons are oxidizing agent, not reducing agent. That statement is wrong. Now let's go to the third answer. NADP plus and FAD functions as coenzymes and electron carriers. Now, what are these coenzymes? Now, coenzymes are non-organic molecules that help in the chemical reactions. They donate electrons, transport electrons, accept electrons. So this is what coenzymes are. So they are saying NADP plus and FAD function as coenzymes and electron carriers. We know both NADP plus and FAD acts as electron carriers, and they are also coenzymes, right? Because they accept the electrons, they transport the electrons, and they also donate the electrons in different places. If you take NADP plus, they donate their electrons during Calvin cycle, and FAD donates their electrons. Actually, the reduced form FADH2 donates their electrons during the citric acid cycle, and that statement is also correct. And the fourth statement, NAD plus functions as an electron carrier and an oxidizing agent. Now, where is this NAD plus involved? NAD plus is involved in aerobic respiration, right? In aerobic respiration, in glycolysis, in both glycolysis and citric acid cycle, NADP plus is reduced into NADH, gaining electrons, right? So NAD plus is involved in aerobic respiration. Okay. FAD is also involved in aerobic respiration, specifically the citric acid cycle. And this statement is also correct, right? Because NAD plus is an electron carrier. They carry the electrons from glycolysis, right, through electron transport chain. And again, they carry electrons from the citric acid cycle to the electron transport chain. Now, final statement, they are saying NAD plus and NADP plus functions as coenzymes and reducing agents. As I told earlier, NADP plus, NAD plus, FAD, these are all oxidizing agents because they accept electrons. That statement is wrong. Now let's go through the second statement. Sorry, the 42nd question. Okay. Some characteristics of organisms and phyla of kingdom fungi are given below. Now, we have three characteristics. We have, first one is cynocytic, multicellular, and unicellular. Now, what does cynocytic means? We know multicellular fungi usually have cell walls between them. Let me draw it. Right now, okay, so okay, let's tell this is a fungi, fungal filament. Now, usually fungi have walls between them, right? Walls, and they have nucleus. So these are called septate fungi. Okay. Now there are certain fungi that doesn't have walls in between them. So they don't have wall. So it is just one long single cell with a single cytoplasm and multiple nucleus. These kind of fungi are called cynocytic fungi. Okay. Now let's go through all the answers. Now, Chytridiomycota is a cynocytic, which is correct. Okay. Zygomycota is also cynocytic, but Ascomycota is not cynocytic. For that reason, we can eliminate the third answer. Why? Because P is going with Z, right? Cynocytic, Ascomycota. Ascomycota is not cynocytic. They have walls in between them. So we'll eliminate this answer. Okay. Now let's go through the other two parts. Now, Chytridiomycota is multicellular, which is correct, and the Ascomycota is also multicellular. So Q can go with both X and both Z. So let me do it the other way, Okay, let me write it down. So what are cynocytic? We know Chytridiomycota is cynocytic, and we know Zygomycota is cynocytic. Then multicellular, we know Chytridiomycota is also can be both multicellular and unicellular, right? And ascomycota can be both multicellular and unicellular. Same goes for unicellular, right? We know both Chytridiomycota and ascomycota can be unicellular. Sorry, something has gone wrong here. Yeah, sinusitic, yes. Ascomycota is ascomycota is not sinusitic, it is zygomycota. Okay. Now let's go through all the answers. Now, first one, P goes with X, correct. Can Q go with Z? Yes, correct. Can R go with Z? Yes, correct. Then first statement is correct. Second statement, P goes with Y. Yes, zygomycota, sinusitic, correct. Q goes with R. Again, what do you mean? Q goes with X. Okay, yes, Chytridiomycota, multicellular. Yes, they can be multicellular, that is correct. And R goes with X. Now, R goes with X, Chytridiomycota, again, they can also be unicellular, that is also correct. Second statement is also correct. And the fourth statement, P is going with X, so X Chytridiomycota, yes, they are sinusitic. And Q goes with X, again, they are also multicellular, and R goes with X, and Chytridiomycota is also unicellular. That statement is also correct. Now let's go through the final statement. Here, they are saying PES goes with Y, zygomycota is sinusitic, yes, it is correct. And the second one, Q is going with Y. Q goes with Y means they are saying zygomycota is multicellular. So this is where the problem is, okay. Now, on the resource book, there is no statement saying zygomycota is multicellular, right? So if you have read about zygomycota in the resource book, they have no mention about whether the zygomycota is multicellular or unicellular, right? For that reason, we really don't know, right? Any student who haven't read more than what's there on the resource book doesn't know whether zygomycota is multicellular or unicellular. So the correct answer is A, B, and D, right? But they have also given marks for the people who have chose E also, which means the fifth answer. Why? Because that's the correct answer, because zygomycota is multicellular. How? It's simple logic, zygomycota is sinusitic. To be sinusitic, a cell usually needs to be multicellular. I hope you get it, right? For that reason, this statement is correct, but they like gave marks for people who even marked it, for people who didn't even mark it. So then R goes with Z. R is it ascomycota unicellular? Again, that is correct, right? So 4 answers of this is correct. But correct answers are 2, A, B, D. For some students marked A, B, D, which means the second answer, right? A, B, D, yes. Sorry, A, B, D, first answer. And A, B, D, E, which means 4 answers are correct, which means it is the fifth one, any other response. Now let's go through question 43. Select the correct statement regarding kingdom Plantae. Reduction in the gametophyte is a trend seen in the evolution of plants. Okay, we know plants have alterations of generations. Plants have sporophyte generations and plants have gametophyte generation. Now they are saying reduction is gametophyte is seen in it's a trend seen in evolution of plants, which means they are saying as plants started to evolve, the gametophyte became reduced. Now let's take a look at it. Okay, now first we have what first plants to evolve was non-vascular plants. Then what evolved? Then it was vasculous seedless plants, right? Seedless plants. Then finally vascular seed plants evolved. We know in non-vascular plants gametophyte is dominant. And in vascular seedless plants, gametophyte is reduced, short-lived, and they partially depend on sporophyte. Okay, they partially depend on sporophyte, but sometimes they are also independent. Okay, they are small, they are reduced, but they can be either partially dependent on sporophyte or they can be independent. Okay, it depends on vascular seedless plants. But in vascular seed plants, gametophyte is microscopic and totally depend on the sporophyte. Scopic. Okay. So the first statement they are saying reduction in gametophyte is a trend seen in the evolution of plants, which is correct because in vascular plants gametophyte is dominant. Then they are small, reduced, and they partially depend on sporophyte. Then finally on vascular seed plants, gametophyte is microscopic. We can't see them at all. So that statement is correct. The second statement, root tissues of vascular plants resemble the sorry. Okay, wait, I'll highlight it first. Okay, they're saying root tissues of vascular plants resemble the stem tissues of early vascular plants. This statement is also correct. This is a statement that is present in the resource book. And from this statement, we can also learn that the root tissues actually develop from the lower part of the stem tissues in earlier plants. Okay, so that's what they are trying to say. Root tissues of living vascular plants resemble the stem tissues of early vascular plants. So which means it means the lower part of the stem actually developed into the roots. And that statement is also correct. And third statement, ancestors of members of kingdom Plantae had key traits of land plants. Now, what are the key traits of land plants? If we take the key traits of land plants, one of the major key trait is the alterations of generation, right? They have sporophyte generation and gametophyte generation. And another key feature is that apical meristems, right? They form roots and shoots. And they have walled spores. What is walled spores? You know, in sporophyte generation, they have sporangium. Sporangium produces spores, and these spores have walls, right? They have a coating. So wall spores. So these are key traits of vascular plants, but they didn't have this in the ancestors. Okay, so this statement is also wrong. So these traits were not present in the ancestors. Fourth statement, members of kingdom Plantae evolved from a group of olive green colored protists. So what are olive green colored protists? Olive green colored protists are sargassum, right? Sargassum is a species that belongs to phylum Protista. But kingdom Plantae evolved from green algae. Okay, they evolved from green algae. We call it chlorophytes. Not green colored, olive green colored protists. So chlorophytes, we also call them green algae. Now if you take this green algae, earlier scientists used to add them to the kingdom Protista, but now they are adding chlorophytes into kingdom Plantae. Okay, also remember that. Final statement, liverworts are evolutionarily closer to hornworts than mosses. Okay, first let's write what phylum they all belong to. Okay, first we have liverworts. Liverworts belong to Hepatophyta, right? So these all belongs to non-vascular plants, right? Liverworts, then we have mosses. Mosses belongs to Bryophyta. Then finally we have hornworts. Hornworts belong to phylum Enterophyta, right? Yes, Enterophyta. Now, this is the order of evolution, okay? So it was in this order. First liverworts arrived, then mosses, then hornworts. So here they are saying liverworts are evolutionary closer to hornworts than to mosses. but livewords are evolutionary closer to Moses, right? Not hornwords. This statement is wrong. Sorry, it's wrong. So the correct answer is, so this statement is also wrong. I don't know why I have highlighted it. Okay, correct statement is A and B1. Okay. Yes. Reduction of gametophytes. Yes. Now let's go through question 44. Which of the following statements regarding life cycle of plant are correct? Again, we need this to solve that question. So let's copy that. It's easy for us, right? Okay. So first statement they are saying gametophyte of Pogonatum is dominant and photosynthetic. I hope you guys remember Pogonatum belongs to phylum Bryophyta, which is a non-vascular plant. And remember, in non-vascular plants, the gametophyte is dominant. Okay, so that statement is correct, and they are also photosynthetic. Second statement, they are saying sporophyte of Salaginella is dominant and photosynthetic. Now, Salaginella belongs to vascular seedless plants, right? We know in vascular seedless plants, sporophyte is dominant and gametophyte is reduced and short-lived. And this statement is also correct. Third statement, they are saying in cycas, sporophyte is dominant and gametophyte partially depends on sporophyte. Now, cycas belongs to Psychodophyta, right? Phylum Psychodophyta. Phylum Psychodophyta is from vascular seed plants. We know in vascular seed plants, gametophyte totally depends on sporophyte, right? Not partially depend on sporophyte. Again, this statement is wrong. In the D statement, gametophyte of Salmonella is reduced and partially depends on sporophyte. Again, this statement is correct. As I told, in vascular seedless plants, gametophyte is reduced and partially depends on the sporophyte. And the final statement is saying gametophyte of Nephrolophis is photosynthetic and partially depends on sporophyte. Now this is where it gets tricky. If you take Salmonella and Nephrolophis, they are both vascular seedless plants. Okay. Now, as I told earlier, vascular seedless plants, gametophyte is partially depends on sporophyte. But another thing to remember, in certain vascular seedless plants, the gametophyte can also be independent. Okay, so mostly if you take so Nephrolophis, they belong to Petrophyta, right? So Petrophyta, what the thing is, gametophyte is independent and photosynthetic, even though they are small and reduced. Okay, remember that. So the correct answer is A, B, and D. Now let's go through the 45th question. Select the correct statements regarding nitrogenous excretory products of animals. Now, before we go through the answers, let's write all the excretory products that animals excrete. Okay. So first we have ammonia, right? Ammonia, very toxic, and it requires a lot of water to be excreted. Then we have urea. Urea is less toxic but requires energy. Then finally we have uric acid. Uric acid, not toxic but requires more energy. Now let's go through the answers. Okay. So the first statement there says secretion of ammonia occurs in human nephrons. So this is where it gets tricky. Okay. We know humans excrete urea, right? Because of that, we think this statement is wrong. But in reality, in the human nephrons, ammonia is also secreted. Why? Is that important? Yes. Ammonia is usually involved in pH regulation of the body. Very, very important for the regulation of the pH. because ammonia, what they do is they bind to hydrogen. Okay, hydrogen ions. What does hydrogen ions do? Hydrogen ions reduce the pH of the body. So what does this ammonia do? They bind to the hydrogen ions and form ammonium ions, and these ammonium ions is secreted out of the kidney, right? So this is a way of our body to regulate the pH, to maintain the pH. So that statement is also correct. But not a lot of ammonia is secreted, okay? Only a very small bit of ammonium ions are secreted from the kidney. Second statement, energy cost for urea production is less than that of ammonia production. Now, ammonia is very toxic, but it doesn't require energy to produce ammonia. But urea is less toxic, but it requires energy to produce urea. So this statement is wrong, because urea production requires more energy than ammonia production. And the third statement, uric acid is the main nitrogenous excretory product of land snails. Yes, this statement is also correct. If you take land snails and then insects, birds, so these all secrete uric acid. And also reptiles, always remember that insects, birds and reptiles, they all release uric acid. Okay, the spelling is wrong. Reptiles. Hurry. Okay, now let's go through the answer. Sharks secrete urea as the main nitrogenous excretory product. Now, ammonia is very toxic. To release ammonia, we need a lot of water, right? We all have learned that to release ammonia, we need a lot of water. Now, they are saying sharks secrete urea as the main excretory product. Yes, this statement is correct, and it's there on the resource book. Sharks secrete urea as the main excretory product. That statement is correct. But let's see why. Let's understand why shark uses urea instead of ammonia. We know uh, the water in the sea is usually salty, right? And urea is also salty. So what does sharks do? Sharks store and secrete urea to regulate that, for osmoregulation, right? To regulate the water content in its body. Because if there's no urea in shark's body, what happens? The shark will lose all the water through osmosis, and it will become dehydrated. For this reason, sharks have mechanism, they secrete urea as a nitrogenous waste product. Okay, that statement is correct. And final statement, they are saying urea is less toxic than uric acid. As I told, uric acid is non-toxic, and urea is bit more toxic than the uric acid. Again, that statement is also wrong. The correct statement is A, C, and D. Right? Okay. Now let's go through the 46th question. In the human brain, 3 ventricles are located in the forebrain. Now, what is the forebrain? Forebrain is the part of the brain that consists of the cerebrum, hypothalamus, thalamus, the pineal glands. Now, here you can see this is the brain, okay? And these are the ventricles of the brain. Always remember that, really important, So you can see, wait, let me, okay. Here you can see this is one ventricle. So this is the forebrain, right? You have the cerebrum here, you have the thalamus, hypothalamus, everything. So this is one ventricle. Then you have another ventricle. Here you can see the third ventricle. So 3 ventricles are present in the forebrain. Now what are these ventricles? So these ventricles are cavities uh, that are present in the brain that is usually filled with cerebrospinal fluid. Okay. Now the brain have total of 4 ventricles and 3 are present in the forebrain. So where is the last ventricle? Here you can see. So this is the last ventricle. Okay, and it is present in the hindbrain, not in the forebrain. So this statement is correct. 3 ventricles are located in the forebrain. So let's highlight it. Now let's go through the second statement. Final body is developed from the embryonic hindbrain. Final body, as I told, belongs to the forebrain part, right? So it is developed from the embryonic forebrain, not the hindbrain. Okay. No, so the spinal body. What is the spinal body? Spinal body is a pineal gland. The spinal gland is usually the one that is involved in the function of the sleep and wake cycle. They secrete melatonin. Okay, remember that also. Okay, this statement is wrong. I don't know why I'm highlighting the wrong statement sometimes. Okay, the third answer, pons varolii is situated between the midbrain and the medulla oblongata. Now, in the brainstem, first we have the midbrain, then we have the pons varolii, then finally we have the medulla oblongata. So this statement is correct because the pons varolii is present between the midbrain and the medulla oblongata in the middle. So this is also correct. Third answer, superficial part of the cerebrum is composed of the nerve cell bodies. Now, we know the superficial part of the cerebrum, but we call this the cerebral cortex, right? And the cerebral cortex is made up of gray matter. Right? Gray matter consists of nerve cell bodies. This statement is correct. Okay. So, anyways, what is the other part? So, inside of the brain, they are made up of white matter. Okay. Also remember the white matter. White matter consists of axons that are myelinated, or myelinated axons, in other words. Okay, this statement is correct. And final statement, hypothalamus is linked to the anterior pituitary gland by long nerve fibers. This statement is wrong. Why? Because hypothalamus is linked by nerve fibers to the posterior pituitary gland, not the anterior pituitary gland. Right? From the posterior pituitary, what happens? Hypothalamus secretes two specific hormones. What's that? Oxytocin and antidiuretic hormone. These two hormones, they travel through the nerves into the pituitary gland and are stored and released. Okay, so it's not the anterior pituitary, it's the posterior pituitary. Now let's go to the 47th question. Select the correct statements regarding the functions of the amnion. So what is amnion? Amnion is one of the four extra embryonic membranes that are present in the fetus, right? So what are the other three membranes? First we have the amnion, we also have the chorion, yes, then we have the yolk sac, finally we have allantois. Okay, these are the four extra embryonic membranes that are present in the fetus. Now, they are saying it protects the fetus from the mother's immune responses. Now, usually this is the function of the chorion, right? Chorion is the embryonic part of the placenta. Okay, so chorion, what they do? They don't allow the mother's blood vessels and the mother's blood and the embryo's blood to mix together. Why? Because the embryo's blood, they contain, or they are also half foreign. Why? Because they contain the father's gene. So they make sure the mother's blood and the child's blood is always separated. So that's the function of the chorion, not the amnion. And second statement, it is associated with the development of the urinary blood of the fetus. Again, development of the urinary bladder is a function of the allantois, not the amnion. Third statement, it helps prevent desiccation of the fetus. Now, fetus is filled with amniotic fluid, right? Now, this amniotic fluid, what they do? They hydrate the fetus. So, it help prevent desiccation means help prevent the fetus dying out. So, this hydration mechanism helps prevent the desiccation, which is correct. So, let's highlight it. Okay, wrong. Let's go through the D answer. And the fourth statement creates a fluid-filled cavity to absorb shocks. Again, the amniotic fluid, it protects the embryo. It protects the fetus. How? It protects the fetus from external pressures or external any harm. If the mother falls, it will protect the mother. It will protect the fetus. And it also any movement of the mother, right? So these fluid, what they do is they absorb the shock and protect the fetus. That statement is also correct. Final statement, it is the source of primordial germ cells of developing gonads. So it is not the amnion, it is the yolk sac, and yolk sac is the source of primordial germ cells of developing gonads. So the correct answer is the C1 and the D1. Okay, now let's go through the 48th question. Transcription process of polypeptide synthesis. Now, when does transcription occurs? Transcription is a part of protein synthesis. Protein synthesis is the process of making new proteins that are required for the body. Now, protein synthesis has two processes. They have transcription and translation. Now, what happens in transcription? In transcription, an mRNA strand is formed using the template DNA strand, right? Okay. Now, let's go through the first statement. Begins when DNA polymerase binds to the promoter site. We know DNA polymerase is involved in DNA replication, not in transcription. In transcription, the enzyme that is involved is RNA polymerase. So RNA polymerase is the one that binds to the promoter site. That statement is wrong. Second statement takes place in the cytoplasm of eukaryotes. Now, where does transcription takes place? Transcription takes place in the nucleus, not in the cytoplasm. Why in the nucleus? Because DNA is present inside the nucleus. Again, that is also wrong. Third statement does not involve DNA helicase. Now, what is the function of the DNA helicase? DNA helicase is usually the enzyme that unwinds the two strands of DNA, right? Unwinds the two strands of DNA and separates them, right? So that is the function of DNA helicase. And are they involved in transcription process? No, right? Why? Because the RNA polymerase also has the helicase activity. So for that reason, the RNA polymerase will unwind and separate the DNA strand. Then again, that statement is correct. Now let's go through the D answer. Add ribonucleotides against the template in the 5 to 3 dash direction. Yes, this statement is also correct, right? Because, see, let me show you guys, okay? So here you can see the DNA strand, right? This is the template strand and this is the mRNA strand, okay? They are reading it from 3 to 5 dash direction, but they are making the RNA strand from 5 to 3 dash direction, right? You can see the RNA polymerase is moving this way, okay, from 5 dash to 3 dash direction. So that statement is also correct. What happened? Okay, how do I erase this? Okay, fourth statement is correct. What about the fifth statement? Converts the information in the mRNA to a sequence of amino acids. Now, as I told, protein synthesis has two processes. They have transcription and they have translation. And converting the mRNA to a sequence of amino acids is that occurs in the process of translation, not in transcription. So what happens in transcription? In transcription, the mRNA strand is formed, right, using a template DNA strand. And this mRNA strand, what they do, they move out of the nucleus into the ribosome, and in the ribosome, translation occurs where mRNA is used to produce a sequence of amino acids. Now let's go through the 49th question. That can be seen in the highest altitudes of Sri Lanka. Now, what are the two ecosystems that are present in the highest altitudes of Sri Lanka? First, we have tropical montane forest, which is present 1,500 meters above. Then second, we have wet Patana grassland. So these are the two ecosystems that are present 1,500 meters above. So first, let's write down all the species that are present in these ecosystems. Okay. So these are the species that are present in tropical montane. First, we have Colophina velcade, Cinnamomum ovilifolium, and Heliocarpus montanus. But if we take wet Patana grassland, we have Chrysopogon nodulibabis. The spelling is wrong for nodulibabis. Yes, it's there. Okay. then we have Ardinella villosa. So these all belongs to wet patana grasslands. So Kimberbogan nardus and Thamada tremula, they don't belong to tropical montane forest or wet patana grassland. They belong to dry patana grassland. This statement is wrong. Okay. Then we have Heliocarpus montanus and Messuaferia. Now Heliocarpus montanus belongs to tropical montane forest, you can see here. But Messuaferia, they don't belong to tropical montane forest, they belong to low wetland rainforest. Okay. Then we have Chrysopogon nodulibarbis and Colophyllum valkyrie. Now you can see Colophyllum valkyrie for tropical montane forest, and Chrysopogon nodulibarbis is present in wetland grassland, wet patana grassland. So that statement is correct. Then fourth statement we have Cinnamomum ovilifolium and Ardinella villosa. Now Cinnamomum ovilifolium is present in which is Valkurdu, which is present in tropical montane forest, which is correct. Then the second one we have Ardinella villosa. Now Ardinella villosa is present in wet patana grassland. That statement is also correct. And if you take Terminalia sabula and Imperata cylindrica, they are not present in both tropical tropical montane forest or wet patana grassland. Okay, but they are present in savanna. Okay, so this statement is wrong. So because why? Because they are present in savanna. Okay, savanna, I think it's double N A, right? Yes. Okay, the correct answer is C1 and D1. Okay guys, now let's go through the final question from the 2023 GCE level biology paper. So they asked during secondary treatment of industrial waste water. We know in waste management, right, we have two processes. We have primary treatment and we have secondary treatment. Now what happens in primary treatment? Now the sewage water, right, we know from the household, the dirty water, they enter the sewage system. And from the sewage system, they enter the primary treatment process. In the primary treatment process, very big particles like the sand, the stones, the glasses, the plastics, these are all removed in the primary treatment. And water, the wastewater that is remaining from the primary treatment enters the secondary treatment. So they are saying in secondary treatment, organic matter is oxidized by microorganisms in the trickling filter. Now, we know there are a lot of pathogens and organic matter that is present in the wastewater that enters the secondary treatment. Now what we have to do, what we usually do is we add microorganisms into the wastewater. Now this occurs in two processes. First one is using the trickling filter and second one is using activated sludge, right? So here I have added an image of the trickling filter. So you can see what happens here is what enters the wastewater from primary treatment enters into these tubes. Let me show it. Into these tubes. Okay, and from these tubes, what they have, what happens is they enter into the tubes above, and the water gets sprinkled. Okay, when the water gets sprinkled to this area contains microorganisms, and the microorganisms that present in this area feed on the wastewater, the organic material that are present in the wastewater, and purify the wastewater, and the purified water enters into this collection tube and goes out through the outlet. So that's what trickling filter is. And the second process is activated sludge. What happens in activated sludge is the organic material, not the organic material, the wastewater. The wastewater enters into a tank, and in this tank microorganisms are added. Again, the microorganisms, what they do is they feed on the organic matter that is present on the wastewater. Okay, so this statement is correct. Organic matter is oxidized by microorganisms in the trickling filter. Sorry. Okay, now let's go to the second part. The solid matter is allowed to settle in tanks. Now, solid matter is allowed to settle in tanks in primary treatment, not in secondary treatment. Okay, what happens in primary treatment is the waste. Okay, the waste from the households, they enter the primary treatment process, and they are allowed into a tank. And in that tank, what happens is they are left for hours. Then what happens? The solid materials that are present starts to settle below the tank, sorry, on the bottom part of the tank. So this occurs in primary treatment, not in the secondary treatment. And third one, more than 75% of organic matter is oxidized. That is correct, because in primary treatment it occurs only like 25 to 35% is oxidized, and in secondary treatment 75 to 95% of the organic matter is oxidized. That is also correct. Okay, the fourth statement, methane is produced. Okay, now this statement is a bit tricky. We might think this is the correct answer, but this is wrong. Why? Because methane is produced, yes, of course, using the sludge that is remaining from the primary treatment and the secondary treatment. Now, if you are thinking what sludge is the solid waste that is remaining, okay? And we have something called the activated sludge, which is the microorganisms that are accumulated in secondary process. So all these sludge, what happens is, can be decomposed using anaerobic bacteria and used to produce methane. Now, this will happen, but it doesn't happen in secondary treatment. It occurs in a very different place. They do it in another place, right? So for that reason, we don't involve it in secondary treatment. That statement is wrong. So D is wrong. Then final statement, sludge remaining after trickling filter treatment is decomposed aerobically. Now, as I told, the sludge, right? The sludge is decomposed to produce methane, and this doesn't happen aerobically. This happens anaerobically. Okay. Now, this statement is also wrong. The correct statement is A and C, which means it's the fifth answer. Okay. Now that we have done with the 2023 A-level biology paper, let's meet in another video, hopefully with the 2022 G.C.E A-level biology English paper. Thank you.